Hello, everyone. Today we'll be talking about the effects of tobacco smoke as a carcinogenic agent. This is a topic that is very personal to me and very interesting. Just as far as my personal background goes, this is a topic that has greatly impacted my life as all four of my grandparents had died due to smoking related causes. Three of them had cancers that were related to tobacco use, and one of them died from complications of COPD. Their age range was from 64 to 73 years old, so this is also something that can drastically decrease your life expectancy as well. But this isn't just personal to me, it's one of the leading causes of death in the country, and it's probably something that has infected each and every one of us. So just to go ahead and frame today's talk, today I'll be starting off with some tobacco smoking statistics to try to help frame the general scope of this topic. And then, because I find it very interesting, is to go over the history of research pertaining to tobacco use. So trying to figure out when did we start to suspect that smoking and tobacco use had detrimental effects. And then I'm going to start going over the mechanisms of tobacco as a carcinogen, trying to get through some of the pathways and some of the effects of how it causes cancer. And of course, we're going to go over some of the limits of our understanding. So just to start off with some smoking statistics, some general statistics: cigarette smoking causes 1 in 5 deaths in the US yearly. So this is something that probably has affected all of us in one way or another, and is a very large cause of death in our country. 1 in 3 cancer deaths are smoking related, and smoking causes 90% of lung cancer deaths. So as we can see, this is very relevant to our class, as it is making up a very large portion of cancer deaths in America. Smokers are 25 to 26 times more likely to develop lung cancer than non-smokers. So as you can already could tell, there's obviously a correlation between smoking and lung cancer. And every year between 2009 and 2013, about 660,000 people were diagnosed with lung cancer or to cancer due to tobacco use, and about half of those people died. So as we can already see, that these are very large numbers that we are dealing with here. So some types of smoking related cancers, it's pretty much short of all of them. Smoking can cause at least 16 different types of cancers in various organs. So this includes the bladder, cervix, esophagus, larynx, lung, mouth region, stomach, blood, colon, rectum, kidney, ureter, liver, pancreas, trachea, and bronchus. So these include the respiratory tract and also organs outside the respiratory tract. So we'll have to try to figure out, why some of these cancers or organs outside the respiratory tract can be affected as well. But it's also important to note that there is hope and that long-term smoking effects are reversible. When a smoker quits, their risk of stroke drops by half after 2 to 5 years. Their risk of non-lung cancers drops by half after 5 years, and their risk for lung cancer drops by half after 10 years. So even if you've been a lifetime smoker, it does drastically help you to quit smoking whenever you can. Also, one study noted that quitting at the point of diagnosis of cancer that is smoking related increases odds of survival as well. So there's tremendous benefits that could be had for quitting smoking. So trying to understand some of the history of understanding of smoking being a carcinogenic agent. It starts way back in a little bit early 1900s, as with the increased mass production of cigarettes and widespread use, doctors started to notice a drastic increase in the incidence of lung cancer. This started to happen from 1900s to the 1940s, but a 1950 population study by Dr. Dahl and Dr. Hill, they took a bunch of data compiled it and were able to find correlations between those that had smoked and then those that had been diagnosed with lung cancer. So as we can see in this chart, we have age of when the patient started smoking and started smoking under 20. That made up a large portion of their population and also 78% of those that were diagnosed with cancer. If we go over one, we see the number of years that they had been smoking, and a lot of them had started smoking or had been smoking for 20 to 40 years, which makes up pretty much almost their whole study. And also the number of years that they had stopped smoking, which a majority of them had not stopped smoking, and that also made up a large portion of their tested patients as well. So just looking at these numbers, you can already tell that there's probably a very good correlation that smoking had something to do with the causation of lung cancer. And as you can see, all of these figures are significant as well. So this study also produced this chart here where on the left we see the death rate from cancer of the lung per million. On the bottom we have years, so starting from 1900 to 1950. And on the right we see the increased consumption of tobacco use and cigarettes. As we can see, the death rate due to lung cancer increased as well as the consumption of tobacco and cigarettes increased as well throughout time. So this didn't give a mechanism, but it did show a nice correlation between use of cigarettes and lung cancer diagnosis. In 1953, there was an in vivo study where experimenters had applied cigarette tar condensate onto the backs of mice. So here they use this apparatus on the left where they would light cigarettes, take their smoke, condense it into an ethanol solution, and then they would apply it to the backs of mice. Of the 81 mice that they had tested, 59% developed papillomas with a mean time of 56 weeks. 44% of their 81 mice that they tested had developed carcinomas with an average time of 71 weeks. And the researchers noted that 71 weeks is about the half-time or half-life of these healthy test mice. So you could correlate that with humans because we usually see about 30 to 40 years of cigarette use before we start to see these detrimental effects, and that could be correlated here with these mice. On this figure down here on the right, we have their percent of lesions that they saw in their tests. And of course, they had the 59% were these papillomas, and it shows it over their mean time, and the 44% of carcinomas. This dashed line shows their control of just them applying the acetone onto the backs of mice, and they had 0 lesions formed. So this was a good gross anatomy study of showing how the use of cigarette tar can cause lesions. 1964 was a big step as far as the nationwide attention to this issue. The first of 2 publications I wanted to cover is a study by Brooks and Lawley, and they were able to determine that hydrocarbons that are called PAHs, which we'll go over later, which are found in cigarettes, could actually bind and mutate DNA. And we see on this chart on the right, going up on the y-axis, we have micromoles of hydrocarbons and how they are able to bind to DNA. And on the x-axis, we have Eyeball's index, which is just an index that measures the carcinogenicity of a compound. So as we're increasing these micromoles of hydrocarbons, we can see that its carcinogenicity is increasing as well. So this helped us give us our first notions of a mechanism behind why cigarettes could be carcinogenic, and that being in DNA mutations. Another major step was also in early 1964. The U.S. Surgeon General at the time, Dr. Luther Terry, had published this large comprehensive review on all the important research that had been done regarding the correlation between smoking and cancer. This helped bring national attention to the dangers of smoking and also concluded definitively that smoking could cause lung and laryngeal cancer. It's still used today as a compiling of a bunch of research that had been done up until the time to see how we in January of 1964, U.S. Surgeon General Dr. Luther Terry published a comprehensive review on decades worth of research that compiled all the correlations. Also in 1964, the U.S. Surgeon General at the time, Dr. Luther Terry, had published a large comprehensive review on many decades worth of research that focused on the correlation between smoking and cancer. This helped bring national attention to some of the dangers of smoking, as it also definitively concluded that smoking could cause lung and laryngeal cancers. Also, it was the first step in the U.S. enacting bills to help combat the dangers of smoking, as in 1969, the U.S. enacted the Public Health Cigarette Smoking Act, which two of the main takeaways from this was it required all tobacco products to have warning labels, as we see here on the right, this 1983 ad where cigarettes can seriously damage your health. And another effect of this act was it banned all advertising on TV and radio for cigarettes and tobacco products. So trying to get into more of the modern day research, what do we know now? Well, tobacco smoke has at least 70 known carcinogenic chemicals in it. Of the 7,000 or so chemicals we know, at least 70 are carcinogens. And these include lead, arsenic, nitrosamines, polycyclic aromatic carbons, and others, which we'll mostly be focusing on the last two for the rest of this presentation. Also, we know that smoking a pack a day of 20 cigarettes causes about 330 genetic mutations per year, or one mutation for every 50 cigarettes. So as you can imagine, throughout the span of a smoker's lifetime, that these mutations can add up over the years. So to help bring this back to the paper that I had y'all read, just a very general information on what we know as far as DNA mutations go. We know that tobacco smoking can add anywhere from 1,000 to 10,000 DNA mutations per cell. Also, the paper noted that 25% of smokers' cells had driver mutations compared to the 4 to 14% of non-smokers. They also noted that longer telomeres protect the DNA from mutations in all subjects. This is why we start to see more detrimental effects as patients get older, because their telomeres get shorter and they can no longer protect this or the DNA mutations. And they also know that there are high levels of variation from cell to cell. So one cell could have thousands of mutations and the cell next to it can only have a few. And this was a chart that was also included in this paper, as we have single base substitutions appear on the top and double base substitutions on the bottom. And as we can see, the control were children, and they hardly had any mutations. And then both ex-smokers and current smokers had significantly higher number of mutations for both single base and double base substitutions than never smokers. And as you can see, there's a lot of variation within this data, but even with their mean data, you can see that it's still significantly higher than non-smokers. So just to get into some of the toxic chemicals and their effects, we're going to mainly talk about two very important chemicals, starting with nicotine and then tobacco-specific nitrosamines, which are derivatives of nicotine, or TSNAs. So kind of just focusing on nicotine itself, it is responsible for the addiction to tobacco products, as I'm sure many of y'all know. Some of its general acute effects is that it can increase heart rate, blood pressure, and vasoconstriction, which, as we'll see, might hint at why it could be detrimental over time. It is also important to note that it is not officially considered a carcinogen, but research has suggested that it can promote proliferation, angiogenesis, and survival. So one study that focused on nicotine specifically, they were able to determine that nicotine can increase tumor vascularization and weight in colorectal cancer in a dose-dependent manner. So as we can see here on the right, we have tumor volume on the y-axis and days after implantation of tumor cells on the x-axis. And as you would expect, the tumor without being exposed to nicotine would increase, the weight would increase over time. But as we can see, after it was exposed to nicotine in both 50 micrograms per milliliter and 200, there was a drastic and significant increase in the weight of the tumor. The researchers noted that this could be due to an increased cellular proliferation via the stimulation of beta adrenoreceptors and also the COX-2 and PGE2 pathways. Now, COX-2 and PGE2 are known to have important roles in colorectal cancer, so this is no surprise as to why those two pathways would be involved in increasing the weight of this colorectal cancer study. Also, they were able to determine that high levels of nicotine were found to increase angiogenesis as well via the vascular endothelial growth factor, or VEGF pathway. This is very similar to the EGF pathway, but is specific to angiogenesis. And they believe that this is also stimulated by beta-2 adrenoreceptors. As we can see here on this chart, the number of blood vessels increase as their doses of nicotine increase as well, but only the 200 microgram assay was significant. It's also important to note, though, that these levels of nicotine that they used in this study were much higher than what we actually see in human smokers. So we wouldn't see 200 micrograms per milliliter in a human smoker. Also, it's also important to note that this study did not account for what nicotine is metabolized into in the body, which is what we'll discuss next. So these tobacco-specific nitrosamines, there's two that are very important as far as tobacco, and they are NNN and NNK. NNN, it stands for N-nitrosaminonicotine, and NNK for nicotine-derived nitrosamine ketone. And these are both derived from nicotine metabolism in the body. So these are considered group 1 carcinogens, which is a high level carcinogenicity, but nicotine itself is not. And the reason that these two are carcinogenic is because they interact with two types of receptors, the beta adrenergic receptors and also nicotinic acetylcholine receptors. And the reason that these two receptors are important, as researchers noted, was, for example, for the BAR receptors, norepinephrine and epinephrine are both stimulators of these receptors. They believe that, for example, NNK is very structurally similar to both norepinephrine and epinephrine, and that is why they can bind to these receptors. So, starting specifically with interactions of our beta-adrenal receptors. So here we have the receptor and our TSNAs bind to it. This can cause a stimulation of cyclic AMP, which can have the direct effect of producing EGF, which will stimulate our EGFR pathway, which we have discussed previously. One result of this is the stimulation of the RAS-RAF-MEK pathway, which I've presented on in the past. And we know that this can cause proliferation as well as other many different effects to DNA transcription. Also, stimulation of RAS can also stimulate PI3K, which will alter the metabolism of the cell and increase its metabolistic capacity. Another downstream effect of this also would be to block BAD, which is responsible for causing apoptosis in the cell. So therefore, we are also inhibiting apoptosis. Additionally, through cyclic AMP, we'll be stimulating these amino acid release of their amino acid metabolite release, which will stimulate the COX pathway, which we've already talked about can have has results where it can affect migration and metastasis, and it's a known metastatic activator. So as you can see, just through the stimulation of this one type of group of receptors, we are upregulating proliferation, we're increasing metastatic ability, we're inhibiting apoptosis, so increasing its survival, and we're also altering its metabolism. Now, for our other set of receptors that it can affect, the nicotinic acetylcholine receptors, we see a lot of the same effects. So once we go through these receptors with our TSNAs, it can cause a calcium influx, which will have a set of downstream effects. One being it can release EGF and stimulate the EGFR pathway. So here we would be upregulating the RAS-RAF-MEK pathway to help increase proliferation. We're also upregulating the PI3K pathway to alter metabolism, which will again inhibit apoptosis through BAD. We are also upregulating HIF1A, which will stimulate our VEGF pathway, which I've already previously mentioned is important for facilitating angiogenesis. So, and then also it's also important to note that this can stimulate the beta-adrenal receptors as well, which is what we just talked about. So here again, we are able to stimulate proliferation, increase metastatic ability, avoid apoptosis, and we're altering our metabolism. So as we can see through both of these receptors, we are covering 5 hallmarks already, with our selective proliferative advantage, altering stress response by inhibiting apoptosis. We're increasing vascularization, we're increasing our metastatic ability, and we're also altering the metabolic processes of the cell. So another compound that is very important to the carcinogenicity of cigarettes are these polycyclic aromatic hydrocarbons, or PAHs. It's important to note that there are 10 types of PAHs present in cigarette smoke, but you can also be exposed to these as well through varying environmental effects as well. The most important that we'll be covering today is BAP, as it's one of the most studied and it's considered highly carcinogenic. And it's able to form DNA adducts, which is just what we talked about in our 1964 research. It just has the ability to bind to DNA and cause mutations. And DNA adduct is just a carcinogenic compound that is able to bind to DNA. And the main mutation that we see are these GC to TA, or just G to T mutations. So as we can see through the study, here on the right, this top pie chart is the lungs of non-smokers, and the bottom is the lung of smokers. And non-smokers will be focusing on this red slice right here, where only 12% of the mutations that they saw were from GC to TA. In the lung of smokers, though, it was 30%. So we can already tell that this is an important part or important specific mutation that is happening in cigarette smokers' lungs. So what are some of the downhill effects of these GC to TA mutations? Well, it inhibits p53, which we know is important for apoptosis. The process follows BAP is inhaled into the lungs. It is metabolized into PDE along with some reactive oxygen species or ROSs. This then binds to the DNA and is able to perform these mutations. And p53 is a hotspot for G nucleotide pairs. So as we are mutating G to T, we're altering the p53 codon, which can inactivate p53 and thus inactivate apoptosis, and we're increasing our risk for lung cancer. However, BAP can do more and is very versatile in its carcinogenicity. It can also stimulate the ERK pathway, which is downstream from the RAS pathway, and which can activate MMP9, which is a known metastatic enzyme. So as we can see here on the left, we have number of invasive cells as we're increasing BAP concentration. And as we can see on all this significant data, and this was performed on gastric cells. As we are increasing our BAP concentration, we are also increasing the number of invasive cells. And you can see this on the right through histology. As we are increasing our BAP concentrations, you can see the marked increase in these invasive cells as well. And this would make sense as ERK pathway can affect hundreds of proteins, including proliferation and metastasis. So some limits to our understanding. So two important general limitations that are pertinent to what we've covered so far is that there is a tremendous amount of variability in both the quantity and location of mutations that can be caused through smoking. As we saw with our topic paper that I gave you, each cell can have its own random number of mutations, and some smokers can smoke for 40 years and not have any type of cancer or tumor, and some do. So it's kind of more of a roll of the dice, but your chances are a lot worse if you are a smoker. Also, there's a lot of disagreement between the levels of chemicals that are tested in research versus levels that can actually be found in smokers. As we previously saw with the nicotine example, they are testing levels of nicotine that were exponentially higher than what we would find in the bodies of smokers. Also, an important limit to our understanding, which is becoming very pertinent to us today, is the increased use of vaping and e-cigarettes. They can contain some carcinogenic compounds, including nicotine, as they can have higher amounts of nicotine than cigarettes do. But one of our largest reasons why we don't really understand it well is because no long-term effects have been yet to be studied, as this is a more recent phenomenon. However, there has been some research that has been conducted, and one study was able to determine that vaping alters bronchial epithelial proteins to the tune of 200 total, and they were also noted that 113 of these proteins were unique to vaping that weren't seen in cigarette smoking. Now, these proteins would have to be studied individually to determine if they have any detrimental effects to any alterations. But this is important to note that vaping can alter proteins in general. Also, another study was able to determine that e-cigarette smoke could induce lung adenocarcinoma and bladder urothelial hyperplasia in mice. So in this study, they exposed mice to vaping or e-cigarette smoke, and a significant number of their test subjects developed cancers and tumors and saw detrimental effects in their tissues. However, in this study, the e-cigarette derived nicotine levels were high, but we aren't really sure how high these levels actually are in humans yet. So that'll be important to see if the levels in humans are higher or the same or even lower than what we see from actual cigarettes. So just some general conclusions. Cigarettes contain numerous carcinogens that can affect various organs throughout the body. So one might affect colon tissue, one might affect lung tissue, one might affect pancreatic tissue. Cigarette smoke uses many of the hallmarks, and I would suspect almost all the hallmarks, but we focused on at least 5 today. And there is a high variance from smoker to smoker as far as the number of mutations that can happen, the actual effects or clinical diagnoses that we'll see from them. So it's important to note that that is relevant. And also, so just some general conclusions to go over. Cigarettes contain numerous carcinogens that can affect various organs. So just like one carcinogen can affect lung tissue, another can affect colon tissue or pancreatic tissue. So this is why we see a whole body effect. Smoking also uses many of the hallmarks, which I would suspect that it uses probably all of them, but we focused on 5 today. There is a high variance from smoker to smoker from what we see from the mutational standpoint, as one smoker can have a lot of mutations or one cell can have a lot of mutations and the other doesn't. So it's important to note that. And of course, vaping will be a very important research target moving forward, and I suspect that it will be proven to be more detrimental than what we think today. Here are some of my works cited. And I'd like to take questions. Thank you.